Hello everyone, my name is Innocent Abel and I'll be representing my article review about the synthesis of titanium dioxide nanoparticle by chemical and green synthesis method and their multifaceted properties. So this is my article review. This is the article that I used. Nanomaterials belonging to the range below 100 nanometer has unique chemical, physical, electrical, mechanical properties and are also diversely utilized in different fields, including medicine, we have agriculture, we have textile, we have in the anti-cancer, drug delivery, and many other fields. So currently, the synthesis of methyl dioxide has been of a very wide application. For example, we have transition metal oxide like titanium dioxide, which is widely used in cosmetics and photocatalysis, medicine, sensor, and solar cell application because of its properties, which are very suitable, like the pores and large surface area. So the interconnected pores and large surface areas of titanium dioxide makes it a very good target. So there's another terminology about the green synthesis. What is green synthesis? So green synthesis is similar to chemical reduction process, but in this method, we use plant extracts to reduce the toxicity, which enhances its biomedical applications. So as we can see from this diagram, we normally have a plant extract and a metallic ionic source. So from this, the plant extract will act as a reducing agent as well as a keeping agent to stabilize the formed nanoparticles. So this will reduce the use of a lot of chemicals. So hence we have good biomedical applications. So this present study investigated the chemical and biosynthesis of titanium dioxide nanoparticles using jasmine flower. So from the plant extracts, we have different secondary metabolites like alkaloids, coumarins, tannins, terpenoids, and saponins. So these phytochemicals are responsible for the reduction of titanium tetraisoproxide to titanium dioxide nanoparticles. So about the methodology, from the materials used, we have titanium tetraisoproxide, we have ethanol, we have methylene blue, as well as distilled water and jasmine flower. So about the synthesis process of titanium dioxide, it involves two different paths. The first is chemical reduction, and the second is bioreduction. So first we can see that we have our starting material, which is normally the TTIP. So then we can go through the chemical reduction process or bioreduction. So once we form the nanoparticles, then finally we perform two tests. The first one is biological activity test, and also the other one is the photocatalytic dye degradation test, which helps about testing about the applicability of our nanoparticles in environmental issues. So for the chemical reduction, 0.1 N of titanium tetraisoproxide is dissolved in 20 mL of ethanol, which is then placed in an autoclave for 3 hours under 150 degrees Celsius. So for the bioreduction part, we just have the plant extract, which is our jasmine flower, as well as the TTIP, 50 mL of TTIP. And then we add the plant extract drop by drop until the color changes towards yellowish gray. So after that, our mixture is filtered and dried at around 110 degrees for 5 hours. So after there, the next step is to do the characterization of the titanium dioxide nanoparticles. So X-ray diffraction is used, FTIR, we use CEM, SEM for surface morphology, as well as UV-Vis for studying about the UV-Vis spectroscopy. So the next is also doing the antibacterial activity, which involves the agar diffusion method. So the culture is incubated at around 7 degrees Celsius for 24 hours under aerobic conditions. So the zone of inhibition is measured to check about the effectiveness of the synthesized nanoparticles. The last part is about photodegradation of methylene blue. So in this process, 100 milligrams of titanium dioxide nanoparticles are mixed with methylene blue in 100 ml of methylene blue solution, and then ultrasonication is performed for 20 minutes. So the absorbance of methylene blue dye is recorded over time. So let's talk about the results and discussion. So from the XRD, we can see that the observed angle at around 27.5 represents the high clustering nature of titanium dioxide nanoparticles. So this is the characteristic of titanium dioxide nanoparticles on the XRD. And also we can see that the green titanium dioxide nanoparticles were exhibiting high intensity due to the presence of polyphenolic compounds in the plant extract. So that's why you see that its intensity is higher compared to the red one, which implies chemical synthesis. So the average clustering size is calculated using this formula, and it was found to be in the range of 31 to 42 nanometer. So the next is FTIR. So through the FTIR, it was confirmed that there was a strong band around 460 between this and 900 per centimeter, which confirms about the availability of titanium oxide titanium as well as titanium oxide vibrations. So the presence of this bond is due to the strong interaction of the kept nanoparticles by the biomolecules, which results in the presence of alkaloids, coumarins, flavonoids, tannins, and terpenoids. As we can see that from the red line, we don't have these groups. But on the green synthesis, we have these groups corresponding to carbon oxygen stretching, which comes, we can see that we have this CO, we have alcohol groups, and CH bendings, which come from the secondary metabolites of our plants. Also, we have the UVV spectroscopy. So the sharp absorbance peak was observed around 385 to 400 nanometer, as you can see. So this confirmed the presence of titanium dioxide nanoparticles. So also from the image of the same, it was revealed that both nanoparticles, for chemically synthesized and biosynthesized, both showed some kind of spherical morphology. And also it was observed that the range of the nanoparticle size is around 32 to 48 nanometer. So in general, the decrease in particle size is inversely proportional to the surface volume of the material. Next is the antibacterial activity. The zone of inhibition layer of the titanium dioxide nanoparticle was examined against E. coli, Staphylococcus aureus, Klebsiella pneumoniae, and the high zone of inhibition layer was observed in green synthesis nanoparticle compared to chemically synthesized nanoparticles. The electrostatic interaction exists between positively titanium dioxide nanoparticle and negatively charged cell wall of the surface of the bacteria, which leads to high inhibition region. As the surface area of nanoparticle increases, there is an increase in surface oxide ion concentration, which results in a more effective destruction of the bacterial cells. As it can be seen, this is the green synthesis, and this is the chemical synthesis. Next is photocatalytic activity. The biologically synthesized nanoparticles have high degradation efficiency compared to chemically synthesized nanoparticles. The degradation efficiency increases due to the presence of hydroxyl group in the jasmine flower extract. In general, when titanium dioxide nanoparticles undergo a UV visible interaction, what happens is that it results in the formation of hydroxyl groups as well as the superoxide groups. So both of these groups are responsible for the degradation of the methylene blue. As it can be observed from these plots, on the plot of green synthesis, the peaks are not too high, are not too tall. So we can see that the height of the peaks is shorter compared to the height of the corresponding peaks, which implies higher effectiveness in this process. So by conclusion, in this present work, titanium dioxide nanoparticles are successfully synthesized by green synthesis and hydrothermal method. The photodegradation of methylene blue under UV visible irradiation results in the degradation of methylene blue to liquomethylene blue. Biomediated titanium dioxide shows maximum degradation efficiency, which is around 89%. The same image reveals that a uniform spherical shape surface morphology was available. So lastly, the biomediated titanium dioxide nanoparticles exhibit a good potency on both antimicrobial and in environmental issues, including wastewater treatment. That's all about my presentation, and thank you.